Good morning. See if we can see me over this podium. It's my pleasure to kind of introduce this first session about leaf traits and how they might be used in remote sensing. I have a kind of a list of an outline of my presentation to first talk about the rationale for why this might be useful. Talking about mapping plant biomes and communities and plant functionality. The second topic I'll go into is relationships between plant species and their spectral signatures and their chemistry. Then the relationship between leaf traits and productivity strategies. I'll mention the TRI database, but obviously Jens is going to talk after me, so I'm going to assume he's going to talk in more detail about this very wonderful database. To look at how do leaf strategies carry over into other plant organs like roots and stems. Lastly, then the remote sensing of these leaf traits with some examples from my lab, but lots of other labs as well, and a conclusion. So one of the reasons I think we would like to try to do this is that current global vegetation maps usually include a small number of community types or biomes on the order of 10 to 20 or maybe 30. And then those units are the characteristics of those communities and biomes are averaged, and you don't see the variation within them, and also obviously inclusions of other groups within them. So if we could work from a plant trait base that's related to their productivity and their physiological functions, we would have a better chance of capturing the carbon and nitrogen and water cycle information. We can't really do it directly from plant species. I found that the Missouri Botanic Garden estimates there's about 400,000 known plant species, so that's a little bit too big. And so somewhere in between, and traits seem to be a way of organizing the information that can be more directly used. So to close the carbon budget, the biggest area that's remaining really has to do with understanding how plant functions affect both the use of carbon dioxide from the atmosphere as well as the feedbacks to the climate system. So if we can't move beyond the state we're in now, we're still quite far from being able to close the budget. And here I show an example of making the spectral measurements. Let's see if this thing works. On the left, then classified this landscape into different functional types, and then applying a relationship to, in this case, to nitrogen, to look at different physiological conditions. The other aspect is to try to understand how disturbance systems affect, alter the carbon and water fluxes in a community. And here I show one from Southern California that John Gammon, David Fuentes et al. Did several years ago, where the ecosystem, you can see the fluxes of carbon dioxide change dramatically, both spatially and temporally. This is from the spring through the summer. We have a very strong summer drought period, although this year we have no rain, so we have a very severe drought right now. Through this fall, and then usually it rains over the winter, and in the spring it's wet again. So these patterns of both source and sink patterns are changing both spatially and temporally. And then at sometime between March and September there was a fire, and then of course everything burned off, and then the system started over again. And then this shows for water vapor flux. So we have, how do you account for those properties when you're averaging. Some work by Greg Asner and Robin Martin. They have been studying the tropical forests for some time, measuring chemistry and leaf reflectance. So here's a list of species that they measured, and these are just the nitrogen and phosphorus relationships. But if you add all the chemistries, they're much more heterogeneous. These tropical species are more chemically diverse than temperate ones. And then this little clustering diagram here shows some relationships between different groups of these plants, but this is their spectra color-coded by wavelength, so from 400 to 2500 nanometers across the visible, and then near infrared and the shortwave infrared. And what you see by the color patterns is that they're quite diverse spectrally as well. So the idea is that you can capture that information in the spectrum. One of the papers that has had a big impact on the idea of using plant traits as opposed to functional types. Why not functional types? Well, you can have a deciduous shrub or an evergreen shrub, but that doesn't mean their light harvesting strategies and their nutrient relationships are the same. So, within those groups, and so you really like to be able to pull them out. And what this paper by Ian Wright et al. Showed was that there were very strong relationships between temperature and rainfall, and here the leaf longevity. So that would go from leaves that are deciduous and just last for a short period of months to leaves that last a year or multiple years. And then this shows for rainfall and two plant properties, leaf longevity again, and leaf mass area. And leaf mass, pretty much all of the traits work better on a mass basis than on an area basis. And that turns out that's really good for remote sensing because the signal we detect is much stronger in the mass basis than it is on an area basis. Here you see that longevity goes up with when rainfall is lower and when the temperatures are higher, and it goes down when the length of the leaf lifespan goes down when there's more rainfall and more moderate temperatures. And then this shows the same thing, except that now you see leaf mass area also has the same relationship. So at the global scale, I think that leaf traits are better descriptive of the economics of plant functional types because of the variety of growth form and history and niche space affect these either the traits or the functional groups somewhat differently. You have a mixture of physiological causation and competitiveness kind of bounds this trait space that result in suites of traits going together. So if you have high productivity, you have high chlorophyll content, high nitrogen, phosphorus, low leaf mass area, and short-lived leaves generally. So you have these kind of traits that go together, which gives you something more than just, say, greenness as an indicator, because you can use the fact that you have multiple traits that should have the same trend to support higher, low productivity kind of environments. And the traits that Ian et al. Identified in that paper that seemed to be held up, that leaf mass area turns out to be really important, and I'll show you that we can, in a number of cases, derive that from the remote sensing data. Longevity just comes from having multiple image captures. Assimilation capacity is it's optimized for light and water and nutrient budgets. So for a continuum from low to high assimilation of rates, leaf nitrogen represents the molecules that are associated with photosynthetic machinery, like rubisco. So if you have a high leaf nitrogen concentration or content, you also have a high potential for photosynthesis. Phosphorus, more or less the same thing, except it's associated with nucleic acid and energy molecules like ATP and so on. These two are highly correlated, and they usually have the same slope in a lot of data sets. Anyway, phosphorus gives insight into biogeochemical cycling, because it's limited mostly by availability in the soil, and dark respiration, which of course is the hardest thing to derive. In this example, which also comes from the same paper, it just goes on to show other correlations between leaf longevity and leaf mass area, here with respiration on a mass basis, or leaf mass area and nitrogen on a mass basis, and phosphorus and nitrogen and leaf mass area. So these are highly correlated traits, but you find that if they're they don't have the same correlation. It's more of a scatter plot if it's on an area basis. This work was extended by this TRI database, which is a huge database with, I think, about 70,000 samples in it, or at least it was in this paper. Oops. In 2011. And you see there's quite a wide range in specific leaf area and in leaf dry mass, but not when you organize it by growth form class, by herbs and trees and shrubs. You see there's a lot of overlap, and it would be rather hard to use these trait or these functional groups to identify what the traits are. It seems better to go the other way around. There's some papers that show that other plant organs have a strategy that's coordinated with the leaf strategy. It makes sense if you're going to have high photosynthetic rates or high rates of transpiration. You have to have a lot of roots for water uptake and nutrient uptake. So you would expect potentially for them to be correlated. And this paper looked at 40 subarctic species and found quite, well, at least the slopes are more or less the same, and they concluded that they indicated they were correlated. Other papers have not found the relationships as strongly. At least they find some things correlated between the traits between the leaves, the shoots, and the roots, but not necessarily according to these leaf economic traits. All right. Just I see too. Oh, there's I knew I heard for a minute. I didn't had a different conclusion, but it seems that the strategies for how plants are going to require and use resources, light harvesting, nutrients uptake and availability, and water, should be coordinated. Not too long ago, this paper by Homolova from looked at estimates of predicting nitrogen, phosphorus, and leaf mass area from plant species from remote sensing data, and found that both on an R-squared basis and on RMSE basis there was pretty good correlation in the prediction, although the strength of the prediction varies. Like the leaf mass area in this one is the R-squares vary quite a bit, and the numbers on the left have to do with how many accuracy determinations that were these are two of them, right? R-square and RMSE, but of the papers, how many accuracy determination there were, and then out of how many papers. And so there's been a number of papers looking at some of the detection of these properties. I would say there's the list of what we probably can detect. Deal with foliar pigments for chlorophyll and carotenoids, water content for sure, dry matter, and leaf mass area, and nitrogen, perhaps phosphorus. Phosphorus is pretty tightly correlated with nitrogen, so perhaps it's not necessary. And then, of course, we have leaf area index on the canopy structure. I think we have to do more with understanding the three-dimensional structure, or some aspect of it, to account for the scattering processes in the spectral information, and indicators of stress, dry biomass and water and pigments, how they change with environmental conditions, and so on. For some examples of retrieving some of these things, I have some examples from a variety of different environments around they're all around the U.S. In the eastern U.S., a temperate deciduous, probably forest, conifer forest in the west coast in the Cascade Range. Some chaparral, some shrub-dominated communities in coastal California and Southern California. Sky oaks is another one, and Los Alamos, which is a woodland and a shrubland dominated system. And then we have some conifer forest, mixed conifer forest in the Sierras, and an orchard example. So I'll start with the orchard example, looking at detecting canopy water content, and this is a paper by Chang et al. Last year in Remote Sensing of Environment. But we had 2 flights over the site in the morning. Each of these is about 3/4 or so of a kilometer on a side, each of these blocks. So there is quite a large fields, and most of the blue, and these are almond orchards, and almonds become the most widely planted crop in California, and also it's most valuable. And then these are younger pistachio trees, and this shows the water content in the morning at 10:50, and then about 2 hours later at 13:05, and we tried to look at the change in water content, which was about 0.54 kilograms per meter square difference in water. So a very small difference in water content, but we're at the point where we can actually co-register the data, count for the BRDF effects and everything else, and come up with a small difference that relates to the diurnal change in water content of the canopies over the day. In this example by Ray Coakley and others from USGS, they looked at what happened after fire and characterized them by their spectral properties. And you see a map showing, this is the color infrared showing the fire scar, and then they mapped it into different surface conditions. If it was ash and charcoal or mineral, the soil been burned off to the mineral soil. If there's dry vegetation, dead straw, or some remaining green vegetation, and looked at the heterogeneity, the patterning of the fire within the burn scar. And they did this by using the absorption features here you see for a laboratory measurement of a leaf with a low lignin level, and one on the dash line with a higher lignin level. And then you see the same thing with the average data. The solid line is the low lignin to cellulose ratio, and the dash line is the high lignin to cellulose ratio. And you see we can match these pretty well. In the This is the Jasper Ridge at Stanford's Biological Field Station, so in the south of San Francisco. And it's a heterogeneous community with different soil types and a combination of forest, shrubs, and grasses in patches. And we looked at a number of variables related to how the plants were using water. So you see we have foliar biomass and dry matter and water content and fuel moisture content and equivalent water thickness and so on. This is a paper that was revised and returned to RSE, and so we haven't heard if it's accepted yet. We had data from 3 different dates, May and spring and fall and September and a late summer date. And so we should be able to capture some of the phenology of these species over this period. And what I wanted to say about it, is that the thing that stands out most is that this shows a relative RMSE, so it's the RMSE term divided by the mean value of the parameter. And we looked at a lot of different indexes, both standard ones like NDVI, but some optimized ones, as well as later I'll show you some data from Prospect, a radiative transfer model. And you see that the error terms for the same indexes are much larger. We have the largest variability in the grasses and the least in the forests in pretty much every case. That was sort of surprising to me, but but the other point that I want to make is that a number of different methods pretty much give the same accuracy. So that I thought that's a little helpful. Here you see foliar water content and leaf area index. Again, the shrubs and the forests tend to have very low RMSE errors. Here's one, leaf mass area, where the shrub community had much higher errors than the grasses or the forest. But in any case, the accuracy of the retrieval is reasonably good. When we applied ProSale, this was with Prospect 5 and I'm sorry, I can't tell you which version of the SAIL model, but it's from last year or so. It's the more recent linking of these. We found that unless you could really assign the correct leaf area index in the soil spectral properties, you couldn't get very good results, very consistently good results, and they were tended to be higher. This just shows for if you let the parameters are free, you always had, pretty much always had the highest rates. If you fix the soil to the correct reflectance, then you did much better, and sometimes the leaf area index. But in general, we didn't do any better with this radiative transfer model than we did with particularly the optimized index, using Prospect to find the best indexes for the properties. Seem to be just as good as method as any. Here you see the pigment retrievals that were tested for Prospect 4 model, and for this shows for chlorophyll equivalent water thickness, which is the volume of water, and leaf mass area for several different databases that were tested, and for separating chlorophyll and carotenoids. So this would be total chlorophyll A and B, or total carotenoids. So this includes all carotenoids, and there's pretty good correlation for both of these. That's all I wanted to say about that. I did want to point out that there's quite an active in the plant biology research community. There seems to be quite active interest in the phylogenetic relationships for these leaf traits. And I just picked this one out as an example where they looked at leaf mass per area across the major clades of plant communities, and you see this, between gymnosperms and flowering plants, there's a general trend across a lot of these that account for some of the variability in leaf mass area, but there is quite a wide range across all plant communities. Both the ecosystem conditions and the species are affecting these traits. Here I show for 3 traits for NDVI and EVI for greenness, and this one for water content across. You can't really see the gray bars, but looking at 2 types, the same communities, but in coastal and inland, or with two different groups. So in the gray is this group of shrubs and grasses, and in the black is a different group. And what, if you could see the gray bars, what you would see is that in some cases the communities do map out the same, but in other traits, which I'll show you in a minute, the inland and the coastal communities, even though the species composition is the same, have quite different responses. For instance, this one where some of these, the overall pattern is more or less the same, but the values of the traits are quite different, and that would have to do with the probably different phenological stage because of the difference in the environmental conditions. So you have both of these things working. And here you see a PRI, which is a photosynthetic reflectance index. It's an indicator of the efficiency of photosynthesis. And like for instance in this index, the same communities, but their functioning, their phenology is quite different over time, whether they're coastal or inland. In this example, I want to stop in just a second. This is we looked at what makes an invasive species successful. In this case, spectrally determined properties. So we looked at chlorophyll content for natives and non-natives, and carotenoids and anthocyanins with pigments. And we looked at the PRI index and how it relates to the isotope ratio. And these invasives seem to have facultative C4 photosynthesis. That's one of the reasons they're more successful. So by going through these different traits, you can see very clearly why the non-natives, the invasive species, were doing much better in the habitat than the native species. And here's an example from the field work. These are where a little section of the Sacramento Delta where all of these, both the native and the invasive ones, were present. And you can see where some of our field data was, and then the modeled relationship for these species. And again, Greg Asner has been working for a long time now with mapping human tropical forests, and here's an example from this paper he did in 2011 with over 6,000 species, and you see the great variability in reflectance and transmission. And he looked at the detection of a whole bunch of different chlorophylls, leaf mass area, nitrogen, lots of different elements with both the full spectrum and just the visible near-infrared spectrum. And then the colored symbols have to do with adding some noise. So the black is the original data, and then the open circles and the diamonds are added noise. So you can see that the R square goes down as you add more noise, and this is the air term. But you, in pretty much every case, you have higher R square and lower RMSE if you have the full spectrum than if you're dealing with just the visible near infrared. And then this just I meant to cut that out. Anyway, this shows for the full data set of leaf properties and chemicals. So with that, I just want to say that the traits are related to leaf processes and their state, and it looks like we can measure most of the ones that were identified in the leaf trait literature. And there's still some primary issues that are unresolved, like the influence of structure on the spectral properties and how that affects the detection. And I would like to stop because I'm out of time, and if you have one question. I think if we don't have any questions, we should go on to Jan's presentation on the TRI database.